In this session, we'll be learning about neuromuscular transmission. So first, let's talk about the overview of muscle contraction. So excited neuron first will act on muscles, and then there is a generation of the impulse or action potential in the muscle. This action potential will later lead to the muscle contraction. So the first step of the muscle contraction is neuromuscular transmission, and that later will lead to the muscle contraction. So that's why it is known as excitation contraction coupling. Motor unit and neuromuscular junction. A single motor unit consists of a motor neuron and all of the muscle fiber that it controls, while neuromuscular junction is the point of synaptic contact between axon terminal of a motor neuron and the muscle fiber it controls. So here in the diagram you can see this is a single motor unit supplying four of the muscle fibers, and on the right side this is another single motor unit that supplied single motor neuron supplies the five of the muscle fibers. Now the structure present in the neuromuscular junctions are presynaptic membrane, that is nerve terminal, synaptic cleft, and postsynaptic membrane, that is motor end plate. This presynaptic nerve terminal membrane consists of lots of mitochondria. It consists of the vesicle containing neurotransmitter, that is acetylcholine. It is also covered by the glial cell, that is Schwann cell, and in the nerve terminal membrane it is consisting of large amount of voltage gated calcium channel. It is separated from the muscles via synaptic cleft, and in the postsynaptic membrane, which is the muscle end plate, it consists of the junctional folds studded with many acetylcholine receptor, in which later acetylcholine binds to this receptor and bring about the neuromuscular transmission. So basically this is a chemical synapse which is cholinergic chemical synapse and is unidirectional in nature, and it is always excitatory, means nerve excitation will later lead to muscle excitation. Acetylcholine vesicle and acetylcholine receptor. Vesicles are actually synthesized in the cell body, moves in exoplasm to nerve terminals via microtubular highway system. Acetylcholine is synthesized in nerve terminal and is taken up by these vesicles. The release of acetylcholine is said to be quantal release. After release of acetylcholine, the vesicles are recovered by nerve terminal cell and again then refilled. While at the junctional fold there is a presence of acetylcholine receptor, which is ionotropic receptor. It has got 5 subunits and its ligand gated ion channels. The type is nicotinic type. As acetylcholine binds to these receptor, a large inward sodium current occurs and a small outward potassium current occurs, thereby resulting a net inward current, and thus depolarizes the muscle end plate and then leads to the excitation of the muscle. So this is the steps of neuromuscular transmission. Propagated action potential reaches the nerve terminal, will lead to the opening of voltage gated calcium channel, and there will be the entry of calcium from extracellular fluid to the intracellular fluid of the nerve terminal. This will help in docking event of the vesicle to the nerve terminal membrane and thus releases the acetylcholine as quanta or package. Now the acetylcholine from the synaptic cleft binds to the nicotinic receptor, will lead to the influx of large amount of sodium while there also occurs the small amount of potassium to the outside of the muscle end plate. So net there is a current is depolarizing current which is known as end plate potential, and this will lead to the muscle contraction. Later acetylcholine is break down to choline and acetate by the enzyme known as acetylcholine esterase present in the junctional fold. This choline is reuptake by the nerve terminal and acetylcholine is resynthesized and packs into the vesicle. Resting membrane potential of skeletal muscle is minus 90 millivolt and threshold potential for the excitation of the muscle is minus 50 millivolt. So this is again the sequence of events during neuromuscular transmission. Motor neuron action potential propagate to the nerve terminal will open the voltage gated calcium channel leading to calcium influx that helps in docking event of the vesicle to the membrane, and acetylcholine is released as quanta. This binds with the nicotinic inotropic receptor, leads to the loss of sodium entry and generates the local potential known as end plate potential. This end plate potential via local current will lead to the opening of the large amount of voltage gated sodium channel, which leads to the influx of sodium to the muscle, and this will lead to the generation of the action potential, and this action potential is propagated and later leads to the muscle contraction. So 60 to 125 vesicles are actually activated from each stimulus. Approximately 10,000 acetylcholine molecules are released from vesicles, while junctional fold of the end plate consists of 15 to 40 million receptors per junction. So this is the last slide showing the agents affecting neuromuscular transmission. Botulinus toxin, it blocks the acetylcholine release from presynaptic terminals, so leading to total blockage and paralysis of respiratory muscles, finally resulting to the death. Curare, it competes with acetylcholine for receptors on motor end plate that will decreases the size of end plate potential. In maximum doses produces the paralysis of respiratory muscles and death occurs. Neostigmine, which is the acetylcholine esterase inhibitors, this prolongs and enhances action of acetylcholine at motor end plate and leads to the muscle contraction. Hemicolinium, it blocks the reuptake of choline into the presynaptic terminal, so that there is a depletion of acetylcholine stores from presynaptic terminal. So this ends our today's discussion. Thank you.